CRISPR, but it's not a genetic model. And that's really our goal, to begin to apply CRISPR to these many great systems that are non-models in the sense they're not genetic models, but they may be great evolutionary models. The two foci for our project are Amberella, which is the sister to all other living flowering plants. So this could provide insights into early angiosperm evolution. And then our second focus is the genus Tragopogon. This is the goat spear genus in the sunflower family, and it's a great example of recent and recurring polyploidy. And our application of CRISPR to Tragopogon will be the focus today. So now I'm going to turn things over to Pam Soltis, who will give us a little bit of overview of our registration today.
So hi, everyone, and thanks, Doug. Welcome, and we're really excited to share our work with all of you today. So thanks very much for joining us. We thought you might be interested in the number of registrants and a little bit of information about the registrants. At about 11:30 our time this morning, we had 304 registrants, which actually beat the cap for Zoom. We're supposed to have a 300-person cap. So among the registrants, 29% are currently using CRISPR in their work. 43% are not using CRISPR, but are planning to in the near future. And 28% are not using it, but are just interested in the technology. Over half of our participants are students or postdocs. Another 30% are researchers, breeders, engineers, botanists, various sorts of professional positions like that. And 14% are faculty or academic scientists. Finally, we have some geographic information about where our registrants, our participants today are coming from. And we basically have registrants from all of the continents except Antarctica. So welcome to everyone. Now, before I turn things over to Bing, I have a few housekeeping items that I'd like to share with everyone. So first of all, please mute. If you don't mute, we'll just mute you instead because we have that power. Secondly, we are going to be recording the webinar. So your participation will represent your agreement to be recorded should you choose to ask a question. Third, we have set up a Google Doc for you to indicate your name, your institution and location, and to record other comments if you so choose. The URL is in the chat, and I have corrected it now so that it's possible for you to edit, or it should be. And we'll continue to repost that in the chat for those of you who have arrived a little bit later. Also, if you can't access Google Docs, please put your name and your institution and your city in the chat instead, so that we can just see who all is actually participating. And this may also provide some opportunities for people to interact with each other. Finally, we'll have a Q&A after the presentations, and we'll have more instructions at that point about how we'll conduct that Q&A. So hopefully that takes care of all of our housekeeping and our logistics. And at this point, I would like to turn things over to Bing Yang, and he will get us started with an overview of CRISPR. So Bing, take it away.
Can you see the screen? Good.
Yes.
Good morning, good evening, and good afternoon, depending on where you are from. And thank you for attending our seminars. At the same time, I would like to thank Pam to take leading on this project and can enable us to come together to work on the project. At the same time, we can do some outreach activities. At the same time, I would like to thank NSF for funding opportunity for us to be a group or external group work on this technology. My name is Bin Yang from Danforth Plant Science Center, and also joint point with Plant Science Division, Bond Life Science Center at the University of Missouri. This is my email address if you have any question about the CRISPR technology or want to request some of the material which we build and use for your research, just shoot me email, and then I will be happy to cooperate with you or send you the materials or the kit which you'll be able to use in your research program. And first, for the genome editing, basically is dependent on the double strand break, and double strand break could be repaired in cellular the process of non-homologous joining, which can make some deletion or insertion at the double strand break cut site. At the same time, could be repaired directly by the homology repair, which can be insert some of the interest DNA fragment or gene, and this called the gene replacement. To achieve the double strand break, there are three types of the restriction or the nuclease. One is the zinc finger nuclease, another is the tenon. The third one is the CRISPR-Cas system. They are all different, but have the common features. For example, the DNA binding domain or DNA recognition could be engineered or programmed for DNA specific recognition. At the same time, the domain can cause double strand break. For example, FOC1 cause double strand break, and single finger recognize DNA. And similarly, tau effector DNA binding recognize DNA, and then FOC1 with double strand break. And then for the CRISPR-Cas system, the DNA recognition is conferred by the guide RNA. The guide RNA can recognize the genomic sequence, and Cas9, for example, can make double strand break. And this is the typical of the CRISPR-Cas system. You have the guide RNA, which contains the Cas9 scaffold and have 20 base pair specific sequence, which could use to tag the DNA. At the same time, you have the Cas9 could be expressed under strong promoter, and then this guide RNA could be built up with a singly or as a combination with multiple guide RNA called the multiplex genome editing. And for our lab or for our group, we use this two CRISPR or two vector system. We build up the Cas9 in the intermediate vector, and they invert or convert this Cas9 into the guide RNA into the Cas9 expression construct, and have the single construct could express both guide RNAs and Cas9. For the last several years, there's other platform for genome editing, which not involves the double strand break and without templates donor DNA. Those are created by Dr. David Liu's group at MIT. And the first part is the base editor. There's the adenine base editor, there's cytosine base editor. Basically, for this group of the base editor, the Cas9 is actively inactivated and fused with the different aminase. For example, the adenine deaminase or cytidine deaminase. The deaminase could function to deaminate the nucleotide into uracil or isoleucine. And this deamination nucleotide could be used as a template for DNA replication to make the permanent DNA conversion, for example, A to G and C to T. There's a third type of called the prime edit or prime base editor, which the Cas9 or deactivated Cas9 is linked with the reverse transcriptase, and then the guide RNA extended to become of the template for RNA. And in this case, and then the reverse transcriptase could use the guide RNA as a template to integrate the nucleotide change at the target site. And for plant genome editing, basically there's another technology has to be used called the plant tissue culture and transformation. The purpose is to recover the transformed and edited plant cell into whole plant or inheritable plants or edit. So that in that case, tissue culture and transformation has to be used to recover or restore the edits from the base or from the genome editing reagent. There's some application for plant genome editing. One is could use for basic biology. For example, could function as a genetic tool for functional genomics to the larger scale or whole genome functional genomics. and could use for gene and trait discovery, for example, can discover or validate of the simple nucleotide polymorphism variation for gene expression or function. At the same time, could use to recapture of those variations by in the init variety can create the novel germplasm with the precise edits. So basically could use for function analysis and could use for application. So for the application part, could increase the crop productivity and the food production, increase the tolerance to pesticide and increase tolerance to abiotic stress and better manage weeds. Eventually will make health and more nutrient food. During last 10 years, about 10 years, and CRISPR-Cas, or especially CRISPR-Cas technology has been used for, or implied for implants, including 45 genus from 25 of the family. Most of the genome editing tools have been put into the model crop plant species, but most of them are widely used for adjacent sperm families, including some of important crop plants. And once we have those genome editing tools and we can create some media or some breedable crop, what are going to do it? And then will depend on regulatory agency. There are 3 categories currently have going on with the genome editing product or crop product. One is represented by gene editing friendly countries, represented by America, United States, and Australia and Canada. They regard this genome editing as non-regulated article or is not distinguishable from traditionally chemically induced gene plant or crop variety. And then there's another like extremely case is the EU still regard of the genome editing crop as the GMO, which we played a very strict regulation on the edited crop. And then there's the third category, which initiated the discussion how to regulate, how to regard this genome edited crop as the either regulated or non-regulated crop species. And then there are some other countries actually has not started or initiated the discussion about the regulation status of those genome editing crop. Hopefully, if all those countries around the world have the unified the regulation to regard the genome editing or part of genome editing crop as a non-regulated article, that's will dramatically increase the breeding program and stimulate lots of the enterprise to use this genome editing tools. And the last one, I wanted to sorry, look like this linked with the video. Basically, I want like to introduce Xiang to talk about basically the hands-on or basic experience about the genome editing he has attained during the last 5-6 years. Xiang, it's yours. Xiang, you're on mute.
Here we go. So can you hear me okay now?
Yes.
I see a slide. Okay, great.
Good.
Okay, yeah, so I go by Shan, which is my last name. And I'm now a postdoc at the Florida Museum of Natural History. So as we mentioned, there are various CRISPR systems, but the most popular and robust one is CRISPR-Cas9. So that's the focus of my topic. So that is considerations in adapting CRISPR-Cas9 in non-model plant systems. So here's our definition of a non-model plant system. So that is species has no reference or has no plant transformation system, but provides a unique study system because of either economic importance and or research significance. So as Doug already mentioned, we are focusing on, in this project, we are focusing on 2 non-model plant systems, Tragpogon and Umbrella. Okay, so this is our outline of my topic today. So we'll start with a choice of plant species. So based on our survey across different CRISPR studies, we found that most species are herbaceous annuals or biennials with a highly homozygous diploid genome. So if you want to choose a plant species, you might want to think about first the plotted level and the genome heterozygosity. So that means a high plotted level will have an increased workload for CRISPR to edit all alleles. And if you have a genome with a high heterozygosity, one single guide RNA may not be able to target all different alleles. So if there's a wide range of plotted level in your plant species or genus of interest, you might want to start with a diploid representative. In addition, if you are working on a polyploid, you can try to design a guide RNA that can target the conserved sequence of different alleles. So therefore, you can mutant all the alleles at one time. And actually, out of the 45 genera we studied, 10 of them include polyploid species. So it's possible to do CRISPR in polyploids. So second, you might want to consider the growth cycle and physiological characters. So these two are closely related, like the annuals, biennials are usually herbaceous, and the perennials are, many perennials are woody. So woody perennials usually have a longer regeneration time, so which makes it challenging to examine certain traits in the mutants, like the flowering traits or seed characters. So with that, out of the 45 genera, 9 of them are woody perennials. So it's also possible to do CRISPR in woody perennials. And I want to highlight one study. So that is annualization of woody perennials. So in this study, they are studying kiwi fruits. So using CRISPR, they edited the CEN-like genes, which produce early flowering edited plants, so which shorten the regeneration time and foster the breeding process. So just to highlight this study. Okay, so once you have a plant species in mind, you want then find the target gene. So, and this is really based on your research questions, but we have a few recommendations for you. So first, you can use the transcriptome data to identify the target gene sequence and predict the intron exon structure. So in our own study in track program, we actually use the ISO6 full-length transcript and then do the blast searches against the orthologs from genetic model plant systems. So in addition, you might want to consider the copy number of the gene in the genome, and you might want to start with a gene has a single or low copy number. So third, generating a loss of function mutant by introducing a double-stranded DNA break as exons is the most common and straightforward CRISPR application. So if you want to start CRISPR in your plant species of interest for the first time, you might want to get a loss of function mutant. So there are mainly two kind of type of genes. First, people knocking out a negative regulatory genes. Therefore, the mutant has an improved trait. On the other hand, many studies are knocking out a marker gene. So the mutant of a marker gene has an herbaceous phenotype, which allows easy visual screening of the mutant. So one great example is called the phytonal desaturase gene, which is the PDS, and I will show you in the next slide. So again, PDS is a popular target gene. It encodes an essential enzyme that participates in the carotenoid biosynthesis pathway. So PDS mutant has an albino phenotype, so you can easily visual screening the mutants. Like here in Petunia, this is the PDS mutant, and this wild type. And actually, this is the first gene we knocked out in the non-model system track program that we are working on. So this is the PDS mutant in track program, so you can easily find it. And also, it's typically a single copy gene in plant genome, so it's kind of easier to knock out all the alleles of the PDS gene. So yeah, this is a pretty good target gene that you can think about. Okay, so once you have the species and gene target gene in mind, you could start the vector construction. So the components of a CRISPR vector applied in a non-model plant system could be adapted from a closely related genetic model species. So basically, there are two components in the vector. So the first is a single guide RNA, and the second is the Cas9. So I will talk about the single guide RNA first. So if you are trying to use CRISPR-Cas9 for knocking out a gene, you might want to constrain the CRISPR-Cas9 target site to the first few exons, which proceed or located within the genomic sequence, encoding the functional domain. And also, people usually identify at least two target sites, so just in case one of them is not working. So here shows our study in track program again. So in this diagram, the line represents the introns, and the box are the exons. So and the blue lines here are the CRISPR target sites. So you can see here there are two CRISPR target sites, and all of them are located within the first few exons. Okay, another thing is that the spacer sequence. So the spacer sequence is at the 5' end, shown here, of the single guide RNA. So this sequence is complementary to the CRISPR-Cas9 target site, which is required for introducing a double-stranded DNA break by the Cas9 endonuclease. So the length of the spacer sequence is, in most cases, is 20 nucleotides with a range of 17 to 24. And at the 3' end of the spacer sequence, we have 10 nucleotide seed region, like shown here. So this really determines the targeting specificity. So, and that means if there's a mismatch at the seed region, it will severely impair the DNA binding capacity. Okay, so here's another thing. So the Cas9 single guide RNA complex combined to either strand of a gene. So this really confused me, so I want to mention it here. So we know that the PAM sequence of the SP-Cas9 Cas9 is NGG. So an N represent any nucleotides. So for example, here we have double-stranded genomic DNA, and we can find, here we go, we can find a PAM sequence on the 6th strand, that is 5' to 3'. And then we can design a single guide RNA based on the sequence upstream of the PAM, like here, right? And then we can have a Cas9 single guide RNA complex binding to this region. Like for example, this is the GACCA, and also in the single guide RNA, it's GACCA. So in this case, it's the 5', the 20 nucleotide single guide RNA, and then followed by the PAM sequence. So on the other hand, you might see that there's also a PAM sequence on the antisense strand, that is from 3' to 5'. And the complementary sequence is CCN on the sense strand. So in this case, you can design a single guide RNA, which is complementary to the sequence downstream of CCN, like here. So this is how it looks like. So it's a CGCCC, but you have a GCGGG in the single guide RNA. So in this case, you have a CCN followed by the 20 nucleotides. So again, so the Cas9 single guide RNA complex can bind to either strand of genes. So actually, you have more targets for the single guide RNA binding site. Okay. So this is the vector express the single guide RNA. So it's, the single guide RNA is driven by the U3, U6, or U3 small nuclear RNA gene promoters, so which is transcribed by the RNA polymerase 3. So we find that in Eudicot, people mostly use the U6 gene promoter from Arabidopsis. And in the monocot, people usually use the U3 and the U6 gene promoter from rice. So that means if you are working on a Eudicot species, you might want to start trying the U6 gene from Arabidopsis. So, and we also find that in some studies, the researchers use the species customized endogenous U6 promoter, which sometimes shows a higher gene editing efficiency compared to those U6 promoter from the model species. So that is another consideration you can think about. So after promoter is a single guide RNA. So there's two parts, the spacer sequence, 20 nucleotide, and followed by the single guide RNA scaffold, which is 76 nucleotide. So the same RNA scaffold or the camera has been used in most CRISPR studies in plants. So you don't need to worry about this part. They already have the backbone. And then it's followed by poly T termination signals. Okay, so, and then a specific first nucleotide could enhance the expression and stability of the single guide RNA. So that is A for a U3 promoter and G for U6 promoter. So here I can give you an example. Like in this case, U6 promoter drives the expression of the single guide RNA. And actually G is the first nucleotide of the spacer sequence, and it's also match the genomic DNA. So this is a perfect situation. So in another case, you might find here, the first nucleotide is T, which is not the G, which would be ideal. So if you have these situations, you could append a G nucleotide to the 5' end of the spacer sequence. So in this case, it does not match the genomic DNA, but it could enhance the expression and stability of the single guide RNA, which may lead to a higher genome editing efficiency. Okay, so here are a few more considerations. First, GC content of the CRISPR-Cas9 target site. So a high CG content enhances the interactions between single guide RNA and the target DNA, and this will favor the targeting efficiency, but it also might lead to a higher risk of off-targeting. Next, the spacer sequence should not contain poly-T, which is the terminator signal for RNA polymerase III. So lastly, the spacer sequence should not pair with a single guide RNA scaffold with more than 6 nucleotides. This is because it will lead to a formation of stem loop structure, which will impact single guide RNA's binding capacity with the target strand. Okay, so if you are working on a model species or a species with a reference genome, there's actually many resources online that can help you to design single guide RNA and evaluate the off-target effects. Here we show 2 examples here and with their link. So the next is the Cas9 endonuclease. So this the vector express the Cas9 gene. So one thing you might want to consider is codon optimization of the Cas9 gene. So we all know that the synonymous codons, that is different codons encode the same amino acid. And there's also codon usage bias between different species. So different organisms may prefer certain codons over others for certain amino acids. And the codon optimization is that it can improve the translation efficiency by changing synonymous codons based on an organism's codon usage bias. So the Cas9 gene is usually driven by a strong and constitutive promoters. And it also usually attaches to a nuclear localization signal, so which ensures the proper transport of Cas9 to the nucleus. Okay, so we summarize the vector usage across different CRISPR studies in plants. So for example, for the Cas9 endonuclease, there are different types of codon optimization. So the most common ones are those codon optimized for Arabidopsis, human, rice, and corn. So like in this diagram, you can find that in the eudicot, which is shown here, so most of them use the Arabidopsis codon optimized Cas9 gene, so which is shown by the circle. And in the monocot, which is shown here, so many of them use the codon optimized Cas9 from rice and corn. Okay, so you can, based on your species of interest, you can think about which version of the Cas9 you might want to use. So in terms of promoters, so we find that the 35S promoter is most widely used across a different species, which is here showed by the black circle. And in the monocots, many of them use the ubiquitin gene promoter from corn, like shown by the black square. And in the eudicots, many of them use the ubiquitin promoter from parsley. So that is shown by the triangle. So lastly, we have the terminator. So we find that most of the studies use the NOS terminator across monocots and eudicots, as shown by the blue circle here. And also in the eudicots, many of them use the P3A terminator, like shown in the square, blue square. Okay, so therefore we have our recommendations for the Cas9 vector. First, you might want to use a strong promoter like 35S promoter or ubiquitin gene promoter. And also for Cas9, it needs to be codon optimized. That is, for monocause, you might want to use the rice or corn codon optimized Cas9, and in eudicause, you might want to try the Arabidopsis codon optimized Cas9. In terms of terminator, you can use NOS terminator. It has been used widely. And if you are working on eudicause, you could also try the P3A terminator. Okay, so once you have assembled the vector, you might add efficiency. So here, we suggest you to start with the transient assays. So compared with the state transformats, which takes months to get, you can get the result of a transient assay in a few days. And this is actually what we did. So highly recommended to start the transient assay before the plant transformation. So again, transient assay is convenient and time efficient. You can get the result in a few days. And these are the two main approaches of transient assays. So agroinfiltration, like shown here, and the prod plus transient assay, like shown here. So you can use transient assay to target either the endogenous genes or the exogenous genes. So like the GFP reporter system using prod plus. So this is a very efficient system that we used in our own studies in track program. So, and I will talk more about this system in the next few slides. So first, the GFP reporter system uses prod plus. So prod plus are plant cell without cell wall. So you use enzyme digestion to remove the cell walls. As shown here, this is a 2.5 hour after enzyme digestion. And this is a 4.5 hour after enzyme digestion. You can see more and more prod plus are releasing from the cutting edge. So after purification and concentration, so these are the prod plus ready to use for the transient assays. So the GFP reporter system for sure uses the GFP as a signal. So for example, here is a wild type GFP gene, and it can express the green fluorescent proteins, which will emit fluorescent signals and the blue to UV light range. So we also have a mutant GFP genes like shown here. So because for one nucleotide insertion shown by this line, we have auto frame or frame shift mutations. In this case, we won't have a functional GFP proteins like shown in gray color. So in more details, in the wild type, we have the ATG start codon, and this is the sequence. And in the mutant GFP gene, so we have one nucleotide insertion like shown here. So, and as you may already find, there is a PAM sequence AGG here. So, and then we can use CRISPR system to knock out this gene. So again, this is a mutant GFP gene. It is a frame shift mutation because of one nucleotide insertion. And this is the PAM sequence. And then we can design a single guide RNA to target this region, like shown here. So the single guide RNA can bind to specifically this region. And it can make a double-stranded DNA break, 3 nucleotides upstream PAM, like at this position, right after the additional nucleotide. And this double-stranded DNA break could be repaired by the error-prone non-homologous end joining pathway. And one possible outcome is one nucleotide deletion. So in that case, they will restore the functional GFP protein, like shown here, and we'll have the GFP protein expressed. So here's our experiment. So we have a prod plus. It's not really this big, but just for the demonstration. And we have a GFP vectors, and then we can transform the prod plus. And then this is a positive control. So the fluorescence protein will be expressed in the prod plus. So this prod plus under the bright field, but if it's under blue light, the prod plus will show the fluorescent signals like shown here. So next, this is our negative control. So here we have the mutant GFP gene. So you transform the prod plus, and you have a non-functional GFP proteins. This is the prod plus under the bright field. And as you can expect it, there's no signal under the blue light. Okay, here comes the CRISPR system. So now you have 3 different vectors. One expresses the single guide RNA, one is the Cas9, and the last one is a mutant GFP gene. So you transform the product path of all of the three vectors. So if the single guide RNA and the Cas9 vector expressed properly, we'll get the single guide RNA at Cas9, and they will assemble the Cas9 single guide RNA complex, and they will bind to the mutant GFP gene. So as you remembered, so this can introduce a double-stranded break and repair by the non-homologous end joining pathway. And one possible outcome is the restoration of the functional GFP gene. So in this case, we'll have a green fluorescent signals in the product path. So this is the product path under the bright field, and under the blue light, you can find that in some of the product paths, we can see the fluorescent signals, which indicate that the CRISPR system is working. So another cool thing about this system is that you can, based on the proportion of a product path emit the fluorescent signals, to determine the efficiency of the CRISPR system. Actually, in our own studies, we compare the efficiency of 3 different CRISPR systems and find the one with which is the most efficient. Okay, so if you find an efficient CRISPR system using the transient assays, that's great, congratulations. You can now start the plant transformation process. So which is the most challenging part for developing CRISPR in normal systems? Okay, so transformation is the process of introducing and expressing foreign genes in plants. So here we are talking about a stable transformation. That is, the foreign DNA is integrated into the plant genome, and the transgenic plant is regenerated. So in a CRISPR studies, the foreign genes are the Cas9 and single guide RNA. So this process is really challenging. So as I quote the sentence from a great review paper, so this says the main bottleneck of applying genome editing to crop functional genomics and crop improvement is plant transformation and regeneration. So this is challenging for crop species. Don't even to mention the non-model system that we are working on. And also in a more recent review paper, they said the transformation and regeneration steps still represent bottlenecks for plant genome editing. So the process of plant transformation is like this. So first, you want to identify an explant with regeneration ability. So the explant is a part of a plant or tissue used for transformation experiment. So second, you developed an efficient system to introduce foreign DNAs into the explant cells. People usually use Agrobacterium or the particle bombardment or the gene gun to introduce the gene. So lastly, you want to select the successful transformats and regenerate plants from the transformat. So let's start with the explant. So actually, people use various tissues or cells for as explant, like shown here. So in rice, they use germinate seeds. People also use cotyledon, leaf cuttings, even the roots, the petioles or embryos. So all different tissues has been used for the explant. But here are the 2 general suggestions. So first, the explants from juvenile plants usually have a higher regenerative capacity. And also in monocots, so the zygotic embryos have most commonly been used as explants because somatic tissues are usually less regenerative. So once you have the explant, you want to transfer the foreign genes into the explants, right? So, and there's 2 commonly used methods. So, and each method has its own pros and cons. So for example, in terms of the Agrobacterium-mediated transformation, which can transfer a tDNA into the plant genome, it's easy to operate, and usually it integrates a lower copy number of transgenes, which is the vector sequence. However, it can cause negative effects on the plant tissues, like browning and necrosis. So comparatively, people also use particle bombardment, like you fire the particles into the plant tissues, which are encoded by DNA. So this method has been shown to be applicable in a wider range of genotypes within a species. However, this method is relatively expensive, which limited its application. Okay, so once you introduce the foreign gene into the explants, you want then identify the transformats. So the most 2 popular selection agents are herbaceous and chalamasins. And these are the genes express make the transformants resistant to those selection agents. So basically, these genes are integrated into the CRISPR vectors, and therefore the transformants will express the resistant genes, and the transformants would be survive on the selection medium, like the example shown here. So this is a medium containing herbaceous, and only those transformants could be survive on those mediums. So sometimes people also integrate the reporter gene, like the GFP gene, into the CRISPR vector. So in that case, only the transformants can emit green fluorescence. Yeah, so and then it can be easily visualized and selected. And also, you can use PCR screening that you design a pair of primers to amplify the CRISPR components. This will also help you to identify the transformants. And lastly, people also use immunoblot. So like using the anti-flag antibodies if a flag tag is attached to the Cas9. Okay, so once you have the transformat, you want to start the plant regeneration process. So regeneration is a morphogenetic process to a stimulus that results in the production of organs, embryos, or entire plants. So this is because of the deficiency of the somatic cells, and plant regeneration sometimes used interchangeably with tissue culture, which is the maintenance of growth of tissue in vitro. So this is the general process of plant regeneration. For example, you can start with leaf cuttings, and then you have a de novo organogenesis. Like first you have a shoot, and then you have the root. Or you can have the callus stage. So the callus is unorganized cell mass, and then go through the de novo organogenesis or somatic embryogenesis process, and you can end up with the transgenic plant. So when I first started to develop the plant regeneration system in Chalcogon, the non-model system that we are working on, I'm thinking that it's not that challenging, right? Because we already know that if you have a high oxygen concentration and a low cytokinin concentration, you will have root regeneration. And if you have a low oxygen concentration but a high cytokinin, you will end up with shoots, right? And you have high concentration of both hormones, you will have a callus. So this seems pretty straightforward. But I changed my mind after I started the experiment because there are so many variables you need to consider, like the hormones. So there are auxins and cytokinins, but there are different types of them. So the concentration matters, and also the combination matters. And also the treatment time can affect the regeneration result. In terms of salt and vitamins, there are different types like the MS, B5, 6, N6, or woody plant medium salt. So these are all have been widely used. And also there are different types of regenerant agents. And there are some other variables like the pH. Sometimes people add a coconut water into it or the active charcoal. So there is so many variables. So, and because of that, we will have a separate webinar, and we are focusing on the plant regeneration system. So stay tuned about the upcoming webinar. Okay, so it's challenging, but there's some good news. So first, you can adapt the process of plant regeneration from a closely related species. And also, this is what I read from the book. Is that I quote here: the tissue culture is much like good cooking. So if you are like cooking or good at cooking, so that can give you extra confidence about developing the plant regeneration system in your own plant species of interest. So, you know, as I said, you can adapt the process from a closely related species. So we are working on Chalcogon from the sunflower family, and we search the research. We find the research on some other sunflower family species, like the chrysanthemum and purple coneflower. So all these two species have successfully developed the plant regeneration system, and we can use their system and try them in Chalcogon. So especially like I shown here from the chrysanthemum studies, we know the importance of the post-treatment hormones, and from the study from the purple coneflower, we start to use the woody plant medium salt. So I will briefly introduce our study here. So first is the shoot regeneration. So here you can see there are two treatments. first is continuous treatment of hormones. That is 31 days on medium with auxin. So the second treatment is the post-treatment of hormones. So that is you put the explant on the medium with cytokinin hormones for 13 days, and then to the medium without hormones. And you can see obviously, so the post-treatment hormones lead to a much higher shooting rate compared to continuous treatment of hormones. So this is what we learned from the study in chrysanthemum. So in terms of root regeneration track program, so we start with 5 days on medium with auxin and also the woody plant medium, and then transfer the shoots to the woody plant medium without hormone for 10 days. So we have a pretty high rooting rate, like shown here. So basically, we have a complete the plant regeneration system in track program. So it takes in total a little bit more than 2 months. So we start with explant and then shooting and then rooting and then acclimation, and we move the plants to the greenhouse. Okay, so that's about plant transformation. So if you have a transgenic plant, you can start the evaluation of genome editing results. So the gene editing can be determined at the DNA level indirectly and directly. I will talk about the indirect method first. So indirectly, you do not need to sequence the target gene. And this method has a high accuracy and throughput. There are 2 popular methods. First is the PCR, restriction enzyme digestion assay. So in this case, the restriction enzyme recognition site is within the CRISPR-Cas9 target site. And the second one is T7E1 assay, that is using the T7 endonuclease 1. So this is the indirect method. You don't need to sequence the DNA. So directly through sequencing, you can determine the specific features of mutations. There are 2 methods. First is a Sanger sequencing. So you can sequence the PCR products either individually through subcloning or as a whole. The other one is high throughput sequencing, which is more costly. You can either sequence a whole genome or a single or multiple PCR products. So this method can help you to identify rare genome editing events and complicated mutations from chimeric individuals. And also you can use whole genome sequencing to identify possible off-target mutations. So this, again, you can examine the gene editing result indirectly and directly. And I will now talk about in more detail about these 2 indirect methods. So the first one is PCR assay. So here, this is a target gene and this restriction enzyme digestion site, which is shown by the black triangle. So the Cas9 string complex can bind to this region and introduce an indel. So because this is repaired by the error-prone non-homologous end joining pathway. So because of the indels, the restriction enzyme digestion site has been destroyed. So the first step of this assay is PCR amplification. So the first scenario is that we only have the wild type allele with the restriction enzyme digestion site. And then we add the restriction enzyme into the PCR product. And as you can see, we have 2 bands here because it has been digested. So the second scenario is that after PCR amplification, we have some wild type alleles, but also some mutations like shown here. So remember, the mutation has destroyed the restriction enzyme digestion site. Therefore, it cannot be digested by the enzyme, but the wild type can be digested. So in this case, we will find 3 bands here. So in the last scenarios, we only have the mutations and there's no digestion, and you can only find 1 band. So again, so based on the gel patterns, you can identify the mutations and also to study the efficiency of the CRISPR system. So this is a very efficient and convenient method. So the next one is the T7E1 assays. So the T7 endonuclease 1 is an enzyme sensitive to the mismatch sequences of double-stranded DNA. So again, we have the CRISPR-Cas9. So if the gene was edited, we'll have an indel like shown here. So the process starts with the PCR amplification. So like shown here, we have both mutations, like shown here, and also the wild type alleles. And then it was followed by denature, which is resulted in a single strand DNA. And then we can reannul those strands. So like shown here, so we have a heteroduplex, which means one strand has a mutation and the other strand is wild type. So this heteroduplex containing mismatched sequences, right? And then you can start at the endonuclease 1 enzyme. So only those heteroduplex can be digested by this enzyme. So again, based on the gel patterns, you can find the genome editing events. So if you want to know the features or the types of mutations, you have to sequence the gene, right? So the features of the mutations mediated by CRISPR-Cas9 are mostly single base insertions, mostly A and T, and also small deletions like 1 to 50 base pairs. So there are different types of mutations. For example, here, the first one is called heterozygous mutation. So we have a one wild type allele and then a mutation allele. So like in more details, this is a wild type sequence. And in the mutated alleles, we have 2 nucleotide deletions, which represented by -2. So the second scenario is called biallelic mutation. So we have 2 different mutations, indel1 and indel2. So in the example here, one of them has 1 nucleotide deletion, a -1, and the other one has a 3 nucleotide deletion, -3. So we could also have homozygous mutation. So all alleles have been edited, but there's only 1 mutation type, like shown here in indel1. So in this case, we have 1 nucleotide insertion, which is represented by +1. So lastly, we have the chimeric mutation, which means in some cells, we have the heterozygous mutation, and in some other cells, we have the biallelic mutation. So in total, there are 3 different indels shown here. Okay, so in summary, so first we talk about the choice of a plant species. So you might want to consider the protein levels and the growth cycle of the species. And then we talk about the target gene selection. So a good example is a marker gene, which is called PDS, which has a very obvious phenotype. You can easily identify them. And then we talk about the vector construction. There are 2 components, a single RNA and a Cas9 gene. So we have different versions of the promoters and a codon optimized Cas9. You might find the best combination for your own system. And then we talk about a very efficient way to examine the genome editing result, that is the transient assay. So you can get the result in a few days. So we are particularly talk about the PROT+ transient assays. And also, we also talk about the plant transformation, which is the most challenging part of the CRISPR studies. But you can adapt the system from closely related species. And lastly, we talk about the evaluation of a genome editing result. And we have 4 different types of mutations like shown here. Okay, so if you want to get more detail about my presentation, you can check out our review paper published last year. And also, again, so we have an upcoming webinar, so that is focusing on the tissue culture process. And also, we will have a symposium and a workshop at the Botany conference. So we have great speakers and it covers various topics. So you are more than welcome to join us. So that's all about my presentation. Thank you for your time.
Great, thank you, Shan. And thanks to everyone again for coming. And we are certainly willing to stay and entertain some questions, but we also know that some of you may need to leave. So if you do need to leave, please feel free to do so. If you would like to stay and ask some questions, we'll go ahead with our question and answer session. So for the question and answer, we'll ask that you please raise your virtual hand if you'd like to ask a question. And or if you prefer, you can also type your question into the chat and I can read it from there. So I'll just open things up for questions now. I've also put references to both the review article that Shan just mentioned, as well as the application to Trigopogon. I've put those in the chat, and I also put them in the Google Doc so that you'll have references to those papers. It looks like Benita has a question. Benita, please go ahead.
Great presentation. I had a one question. I saw in your presentation that you designed the construct separately for the guide RNA and separately for the Cas9. So my question was like, was it not possible to design a single construct with both of those components?
Yeah, Bin, do you want me to answer the question?
Well, like I can answer that.
Okay,
And
sure.
for CRISPR-Cas system, it's two components. One is guide RNA, another is Cas protein. And Cas construct, you do not need to construct any of the guide Cas9 construct once you have one. So Cas9 construct or expression construct is universal. So every time you just only build up the guide RNA by replace that 20 base pair spacer sequence upstream of this guide RNA scaffold, and then mobilize that guide RNA expression construct into Cas9 construct to form single guide RNA. Single construct could express two components.
Okay, because like when I I did CRISPR based editing like in Peanut during my master's, and I had designed a single construct with both of those components together. So I was just wondering.
Mm-hmm. Yeah, you can use one single construct which already contains the Cas9 expression construct. You don't need to do anything on the Cas9 part.
Thank you.
You're welcome.
Thank you.
Yeah, I
so
want
we-
to add.
Oh, go ahead. No, go ahead, Shan,
Yeah.
please.
Yeah, thanks for the question. Yeah, so actually, we do have one construct, but just for demonstration purpose, I separate them. So I just talk one after the other. But yeah, we do have a single construct expressing both components. Yeah, that makes the system more efficient. That's a good point.
Thanks for your question. We have our next question is from Ashwini Talakayala, who says, I want to know how we can target plant viral genome editing. Is it mandatory to keep the Cas9 every time active, like in transgenics? Bing, do you want to take that one?
Okay. So basically, if you do like antiviral genome editing, there's two types of the viruses, the DNA virus, the RNA virus. So you need to choose different type of the CRISPR-Cas system. There's RNA degraded CRISPR system called the Cas or called Cas13, and you can express this Cas13, this is the Cas13 in the plant. Or if you target the DNA of the or virus DNA and then use Cas9 or CPF1 or Cas12a. And then certainly that's kind of the antivirus, you have always to keep the reagent active. So which means you may want to make the stable transgenic plants which express those components when the virus got in or virus still persists to work.
Thanks. And then there's a follow-up question also that says, if we do breeding, is it possible to get resistance or only proof of concept? If I'm understanding, if I'm reading that correctly.
Well, for this virus, if you could do two ways. First, you need to demonstrate the system work in the in the plants you want to breed, and the second, you want to actually move this proof of concept into the init variety or cultivar you want to use it, and then you want to demonstrate it work. At the same time, you may want to do the field trial at the same time, because you keep express this CRISPR-Cas system in the plant, you may go through regulatory agency which regard of your CRISPR as the transgene or foreign gene. So that may be characterized as a GMO.
Thanks. And Benita has another question. She asks, after protoplast transformation, how do you count or measure the efficiency of transformed cells? Do you use the cytometer?
Xiang, would you go ahead?
Ken, do you want to
Yeah,
take that one?
Sure, yeah. So in short, we do not use a cytometer. So basically, we just have different regions and take multiple pictures, and then count the number of pearl plots in each region, and the number of those have the fluorescent signals, and then you average a few regions. So that's how we count the transformation efficiency.
And I also want to add, if you want to demonstrate, you want to assess the efficiency or activity of the CRISPR-Cas9 system, you want to look at the time of the GFP come out and also the density of the GFP in individual protoplast cells.
Thanks. Are there any other questions? I suppose I should check on the Google Doc as well. Okay.
Well, if some audience wanted to open to go on the video, and also it's time for communication too, and I would like to see your face and do some of the direct, even though we cannot meet in person, I think meet on screen is good too.
Mm-hmm.
You're welcome to basically open your video and ask questions.
Yes, exactly. But we do have another question in the chat. Sumitra, if you'd like me to read your question, I will, or you're welcome to ask it yourself. Maybe I'll go ahead. So Sumitra Paul asks, in one of our experiments, we're screening the GE lines. We found the mutation in the expected region and another site in the same gene, but the second site has no PAM site. Could you please explain? We have checked with restriction enzyme digestion and sequencing of the PCR product. You can probably, it might help maybe Bing or Shan if you guys read it yourselves instead of having, it might be easier to remember all of the parts.
Well, I think probably will be viewers look at that site with unexpected site very carefully, and you may want to really go into your sequencing trace file to look at that mutation is true or is just only the sequence read by the machine or algorithm give you that sequence. So sometimes the actual mutation given by the sequencing facility or the reads from the software may not actually reflect the mutation from the chromatography or the trace sequencing file. So you need to be very careful. But in case there's the unexpected mutation or not related to the PAM sequence, you need to be very careful. Could either be PCR error, could be either by sequencing error, could be off-target mutation. So it depends on what kind of situation you want to look into it.
Thanks. Our next question is from Ahmed Jalilian. How can I find off-target in a non-model plant?
Xiang, you want to go ahead?
Yeah, actually, I don't know the answer. It's very challenging, right? So you don't have a reference genome and you don't know the off-target site. But I'm thinking that you might be able to find some potential off-target sites. That is, the DNA region with a few with a variation of few nucleotides compared to the single RNA. So that might be a good start to examine the off-target effects. But you might not be able to do it at the whole genome level. So Bing, how do you think?
Yes, I think because if you use like unmodeled plants, basically you do not have the reference genome, or you do not know if your guide design is perfect, and if how many of the potential off-target existing genome. So basically it's a black box.
Okay, our next question is from Mukhtar Davis, who asks, in CRISPR genome editing using stable transformation, Are Cas9 and the guide sequence cassettes integrated into the plant genome? Is it possible that the editing event occurs multiple times to the same target site because of the constitutive expression of both Cas9 and guide sequence?
Yeah, I can take this one.
Go ahead.
So yeah, the answer is for your question. First question is yes. So it is integrated into the plant genome. So the CRISPR might continue to work if you still have wild type alleles. And actually, in some polyploid species, in a single generation, you might not be able to mutate or edit all genes. And in the following generations, you will have more additions. But the thing is that if allele has been already edited, the sequence might be changed, and then it will prevent the further addition by the other CRISPR. So I hope that will answer your question.
Yes, I think that's right. On this, because you are selecting the transgenics, which based on the selection marker of your transgene or CRISPR construct. There's a couple of reports that says if you don't select your CRISPR-Cas reagent, you may end up the plant has a mutation or edit, but no CRISPR-Cas because you don't select it. Those DNA construct or DNA will be segregated or will be sorted out during the tissue culture process or degraded or not integrated in the genome.
Hey folks, can I introduce something quickly? This is Doug. I think this goes back to Ahmad's question and looking at off-target in non-model plants. And I think it's important to stress that we do have some genomic resources for Tragopogon, so it's not a total black box. I think Shan mentioned initially that we did have a lot of transcriptome data, and we did have a sort of shallow genome assembly. I think we had, 40–50% of the gene space covered. So, I think that's something else that people can keep in mind when they're beginning a project on a non-model. If you can generate some of these genomic resources, or maybe you already have them, even if there's no other real model system close by, you can, especially now fairly cheaply, get at least a starting point. And maybe Bing and Shan can say more, but I know that those resources did help us a lot in getting Tragopogon off the ground.
Yeah, Doc, what you're saying is absolutely right. And the build up the resource is kind of the gradually accumulation of the knowledge or information, and may not necessarily build up those resources or sequence resource can immediately help your project and also future projects in your lab too.
Thanks. So we had another question from Benita. She asked, do you use software to design the constructs?
So I don't, but I don't know how about in Bain's lab. Do you guys use it?
Yes, we use the software to some screen, some software like show on the slide, and they give you pretty good, especially if you have the genome, knowing the software will give you some of the potential off-target for choice, at the same time give you score, which could use as a reference to choose the stack site.
You are talking about guide RNAs, right?
Yeah, choose the select the tag site for guide RNA.
I was asking for like construct, like fusing all these promoters and Cas and then everything. So do you use like gateways or, I mean?
Well, you could consider two options. One, if you initiate your project, you can read the literature to see if there's available of the system you can request for. And then in that case, that saves your time and energy. And then you can try that system. And the second, if you prefer, or if you do not find the available system, you can build it yourself. So when you build your own system, then you have to go through the process Xiang talked about it. Choose different promoter, optimize the construct or the gene, and assemble them together, or those kind of the molecular biology skills.
Thank you.
So it doesn't look like we have any more questions in the chat. Oh, we just got one. Okay, so let's see. This one is from Nicholas Farrow-Wajaya, and the question is, does ribonucleoprotein for CRISPR-Cas9 have the same efficiency as Agrobacterium-mediated? I'm asking this because to avoid our mutated plant being labeled as transgenic plants, the researcher must use the final product of CRISPR-Cas, which is Cas9 guide RNA complex.
Xian, you want me to go ahead and pick up this one?
Yeah, I would say I don't know. I know there's some people like use already assemble like a Cas9 complex and integrate to the plant genome into the plant cells, and that is not like genome edited plants. But I never done that experiment by myself, and I don't know how would the efficiency be different.
Well, for the efficiency, certainly it's not as efficient as the integrated the construct, because the ribonuclease is the premixture of the guide and the Cas9 or Cas protein. Those complexes cannot exist in the plant cell for a long time, probably several days, and then the mutation or the edit will happen in a very short time. At the same time, you don't have any selection, and basically the efficiency certainly will be much lower than the stably integrated of the CRISPR-Cas system, and which you can select the transgenic plant, and the transgenic plant will have very high efficiency of the edit. And then if you remember, the integrated construct may not link to the edit. So you can segregate out of this foreign or construct in the next generation through genetic crossing and the segregation, and then you got the non-segregant, which contain the edit, not the DNA construct. In the US, they will not recognize those plants which contain edit, but no CRISPR constructs, DNA.
Thank you. Okay, so now it looks like we don't have any more questions. So I think at this point, we will wrap up. But what I would like to call everyone's attention to is the fact that this Google Doc will remain open. And if additional questions occur to you and you would like to jot them down there, we can return to those, and that way they're all posted and can be answered for the group as a whole. Alternatively, if you would like to contact any of us, we'll put our contact information in the Google Doc as well, so that you can contact us if you have other questions. And I think at this point, I'll offer a big thank you to Bing and Shan for their great presentations. And Doug, I'll turn it over to you to close things up.
Oh, thanks, Pam. No, nothing more to say, but thanks to everybody for attending. Right, any questions, please follow up with any of us. And as Shan mentioned, we're going to have additional opportunities for participation in webinars. And as I said at the introduction, hopefully some hands-on experience, as is part of our proposal, our grant that was funded, to provide hands-on training opportunities. Of course, we can't do that yet, but we're hoping that day will come. Maybe this fall, who knows? But stay tuned. Shan, do you want to say anything?
I'm looking for something, so that is great.
Okay. Well, thanks a lot.
Great. Thank you, everyone.
Thanks, everyone.
Thank you.
How, Sean, I can stay for a while?
Sure, yeah.
Yeah.
I think I will start recording, right?
Yeah, that would be
Yes.
great. Thanks.